In this session, we are going to understand the concepts of major histocompatibility complex and human leukocyte antigens. What are the classes of major histocompatibility complex proteins? What is transplantation? What are its types? And what is the importance of human leukocyte antigens in transplantation? And in the end, we will learn about different methods of human leukocyte antigen typing in the laboratory. So let's begin with the definition. There are certain terms which I am going to use in my session. So it's better that we can have an overview in the beginning. So first of all, the question comes at what is major histocompatibility complex? Students, this term is derived from the early research into the acceptance or rejection of the tissues. As you know that this name is comprises of major, which means that most important. Histocompatibility, it means that this complex is related with something which is associated with compatibility. Histo means tissue, so compatibility between the tissues of a donor and a recipient. So overall, a major histocompatibility complex or MHC is a group of genes that encode for the proteins found on the surfaces of the cell, and these proteins help the immune system to recognize foreign substances. It means this group of genes is coding those proteins which are helping our immune system so that they can differentiate between the self and the non-self. Another term which is simultaneously used by scientists in accordance with MHC is human leukocyte antigens. Students, human leukocyte antigens are the group of related proteins which are encoded by the MHC. As I have told you that MHC is a region. It's a region which is comprising of genes, certain genes, and these genes are encoding certain proteins. So those proteins are actually human leukocyte antigens. Sometimes the MHC and HLA are using simultaneously, because in human, and when we are talking about the MHC of human, we are using the term human leukocyte antigens, not MHC. Sometimes we are using MHC more commonly, and sometimes we are using HLA, means human leukocyte antigen. So in short, you can say that HLA is the human body's version of MHC. The word MHC is utilized in every vertebra, from smaller to the larger animals, but HLA is exclusive term for human beings only. Okay, another term is the alloantigens. Alloantigen means those antigens which are present in some individuals of the same species, like the basic ABO blood group systems in humans. We all have that ABO blood group, but somebody is A positive, the another person is B positive, so on. So these are the antigens which are not same in every species, but can be different from person to person. Haplotypes means with the two sets of genes, means that one set is from the paternal chromosome and the second one is on the maternal chromosome. Another terminology is codominant, that the proteins which are encoded by both the paternal and maternal genes. You know that when we say that dominant gene, it means either it is from maternal side or from the paternal side, but codominant means that the genes are inherited from both parents, from maternal side as well as from the paternal side. Okay, so now major histocompatibility complex is, as I've told you, it's the region of genes. These are the glycoproteins, and they are found on the surfaces of the eukaryotic cells, and their basic function is that they help the leukocyte so that the leukocyte can distinguish between the healthy cells or unhealthy cells, or you can say infected cells, or in simple words we can say that in between the cells and the non-cells. The success of the transplant tissue, whether it's a tissue or any organ, it is highly dependent upon the donors and the recipient's human leukocyte antigens, which are encoded by the HLA genes. As I have told you the definition of alloantigens, so we must know that the HLA proteins, they are the alloantigens. Okay, now if a person, means if the donor and the recipient have different HLAs on their cells, this will increase the or stimulate the immune response. And whenever there is an stimulation and the triggering of the immune response, the ultimate result is a transplant rejection. So therefore, a good donor and a good recipient is always, this combination is always created when there is a matching of their HLAs. So HLA genes, where are they in the body? They are located on the short arm of chromosome 6, and there are you can say that different type of MHC proteins like class 1 and class 2 and class 3, but most commonly we are dealing with the class 1 MHC proteins and class 2 MHC proteins. The class 1 MHC proteins are encoded by HLA-A, HLA-B and HLA-C. Class 2 proteins are encoded by the regions DP, DQ and DR. And the class 3 proteins are between the loci of class 1 and class 2. Now let's see in this diagram that what we are seeing, that this is the HLA complex and this is a chromosome 6 with a long arm and the short arm. Now you can see the red color is showing the HLA region, means this is the region where we are having the genes for class 1 and class 2. As I have told you that HLA region has different loci like HLA-A, HLA-B and HLA-C. They are primarily dealing with the class 1 proteins. And there are certain regions, they are given the names by the scientists like DP, DQ and DR, and they are encoding the class 2 proteins. And in between class 1 and class 2 we have the loci for the class 3 proteins. Okay, so you can also get the concept of this HLA complex by figure 62.1 of your Warren Levinson. In between the class 1 and class 2, in this picture you can also appreciate the class 3 where normally which are encoding the genes for C2, C4 tumor necrosis factors and LT. Okay. Now about these HLA proteins and HLA genes, that each individual has two haplotype. It means that two sets of these genes are inherited, one from the paternal side and another on the maternal side. Also these genes are highly polymorphic, means that they have, or you can say that there are many alleles of the class 1 and the class 2 genes. For example, it is said that there are about 47 HLA-A genes and there are about 29 HLA-C genes and about 82 HLA-B genes. And it is said that there are more than 300 HLA-D genes. But you know, it varies from individual to individual, means that a single person inherits only a single allele at each locus from each parents. So therefore, a person, you can say that it cannot have more than two class 1 and class 2 proteins at each locus. Okay, now this was about the major antigens, major MHC antigens, but we have certain minor antigens too. The minor antigens, they also have a very important role, although we do not have the exact number of minor antigens as we have a major antigen, but again they are allelic variants. They are encoded by the genes which are at other sites than HLA loci, and these minor antigens, they evoke, you can say that evoke the response, although or at that not level, you can say that the level is comparatively weaker, but it can cause the transplant rejection at the slower speed. Okay, now let's move to the another part of the presentation, that is the MHC proteins. I am saying that class 1 protein, class 2 proteins, but now let's see that what are the major differences between the class 1 and class 2 proteins. So this is a very basic structure of a class 1 MHC molecule and class 2 MHC molecule. The class 1 MHC molecule has 3 alpha chains, means alpha 1, alpha 2 and alpha 3, and they are linked up with a beta microglobulin. While in class 2 MHC molecules we have 2 alpha, means alpha 1 and alpha 2 chains, and 2 beta, means beta 1 and beta 2 chains. There is no beta microglobulin. So what is the basic difference between the class MHC class 1 and MHC class 2? Let's see. Okay, MHC class 1, it has about 8 to 10 amino acids, and class 2 has about 13 to 18 amino acids. So it means MHC class 2 is comparatively you can say that a larger molecule. The peptide binding domain, means this is a part where the peptides are bind, proteins are bound. They are alpha 1 and alpha 2 in MHC class 1. In class 2 we have alpha 1 and beta 1. MHC class 1, they present antigen to the CD8 T cells, means they are associated with CD8 T cells, while MHC class 2, they are associated more with the CD4 T cells. MHC class 1, they are found on all nucleated cells, okay, while MHC class 2 means that they are found on the surfaces of antigen presenting cells, means mostly macrophages and dendritic cells, and also on the surfaces of the activated T cells. The class 1 proteins, they are composed of one peptide, which is encoded in HLA locus and beta 2 microglobulin, as I have shown you in the picture. But class 2 proteins, they are composed of two peptides, and they are encoded by, again, HLA locus. In class 1 bind with the endogenous antigen, and class 2 are mostly with the exogenous antigen. MHC class 1 has no invariant chains, while the MHC class 2 has the invariant chain. So these MHC class 1 and class 2 proteins are extremely important because you know that overall they are encoded by various loci, as we have discussed, and it is said that about 0.1% of the total human genome is of this by this MHC is encoded by these loci, and comprises of MHC class 1 and MHC class 2. Okay, so I hope that you have got a concept of major histocompatibility complex and the classes of the proteins, MHC class 1 and class 2 proteins. Now let's discuss the transplantation. That what is the transplantation? It is the process of moving cells, tissues or organs from one side to another for the purpose of replacing or repairing damaged or diseased organs and tissues. Okay, so transplantation is of different type. We have autografts, we have isografts, we have allo and the xenograft. Graft is that tissue or that organ which is being transferred from one side to another side. So don't get confused at what is this term graft. The graft and transplant are the terms which are used. You know that sometimes the people are using the word graft, sometimes people are using the word transplant. Okay, now let's move to the first term, that is the autograft. Autograft means that the graft is in the same person, like from one body part to the other area, like most of the time skin grafting, hair transplantation. You know that we are just taking the hair from the occiput region to the frontal region. Sometimes the coronary artery bypass, that is also an example of autograft, in which we are taking the part of the long saphenous vein, most commonly from the leg, and then we are transplanting it into the heart. So these are the examples of autograft, and autograft is never rejected because it is taken from the same person. Okay, so it is never ever rejected. Then we have another type of graft, which is isograft. Iso means same and graft means that the transplanted tissue or organ. So isograft is between the identical twins, means twins should be, so obviously the chances of rejection of isograft are almost always negligible, because the twins have inherited almost the same genes, the same loci, codominant genes from their parents. So therefore they have a very good match between the MHC proteins. Then we have an allograft, which is the most common graft performed these days for transplantation purposes. It means that the transference of the tissue is between the different members of the same species, like from human to human. For example, kidney transplantation, heart, lung, liver, even corneal transplantation. So allografts are between the species of the, you can say, individuals of the same species. Then finally we have a xenograft. The xenograft is between the two different species, like for example from an animal to the human, okay, or between the two different animals, like from a rabbit to the guinea pig, or guinea pig to the rabbit. So the xenografts are almost always rejected until and unless the immunosuppression is not given. Okay, now how the autograft is accepted? As I have told you that autograft means that a transplantation of the graft within the same individual. So what happens, for example, the grafted epidermis, in this picture you can see that there is a grafted epidermis. In this picture A, the blood vessels are broken apart. But after about a week or within 2 weeks, the process of the healing will take place. And what will happen? The resolution. The revascularization in the picture A will move to the process of the resolutions, anastomosis between the vessels, microvessels starts taking place, and finally a complete anastomotic region is developed with proper revascularization, and in this way an autograft is accepted very well by the body. Okay, now what is the role of MHC? Why we are talking about MHC? As I have told you that MHC is very important because it is enabling the white blood cells and the immune cells to distinguish between the healthy cells and the foreign invaders. Okay, so you can say that therefore they have a very important role in allograft rejections, and the process by which the allografts are rejected is called as allograft rejection. So basically the allograft rejection is because of the, or you can say that it is of two types, or it is of the course is of two types. First one is the primary rejection, which is the first set rejection, and this is a T cell mediated immune response. Because of the T cell mediated immune response, the transplant can be rejected within, you can say that within a week, within 3 to 5 days. And then we have an accelerated rejection, which is a second set rejection. In accelerated rejection, this is because of the pre-sensitized T cells, means the T cells which were primarily, they were not sensitized, but now they are sensitized, and this is known as the second set rejection. And it is taking place about, you can say that by cytotoxic T cells. So cytotoxic T cells have very important role in determining that whether the transplanted organ or tissue will be accepted or not. Okay. This is the immunological basis of graft, that how the graft is rejected. For example, just for the understanding the concept, we have taken a green mouse or a green strain mouse and a black strain mouse. So what happened that we took a tissue of the, or you can say that the graft from the skin, and we implanted onto the black strain mouse. So what will happen that initially a primary rejection will take place, means within a week or even more, not more than 10 days, a primary reaction which is known as the first set reaction, and it is a T cell mediated reaction will occur, and it will cause the rejection of the tissues. But what will happen that antibodies will contribute to the rejection of certain transplant, like especially bone marrow. But in the skin we have basically the T cell mediated immune response. So what will happen that now there are two possibilities after the 6 months, means almost a year, when the same rejection can be occur because of the secondary onset, and this rejection now will take not 10 days, it will take about 3 to 5 days. And what happens that again that black strain mouse, if it is grafted by the blue strain mouse, so the primary rejection will again takes place in 10 days. So basically the concept is that the primary rejection, although it is taken about 7 to 10 days, but what will happen that the next time when the same organism is sensitized with the same tissue, the rejection will take place in a shorter interval, and it will take about 3 to 5 days. Okay, so basically the major role in allograft rejection is by the proteins encoded by the DR locus. This is an important point. And also these alloantigens, as I've told you that they are alloantigens, they will activate the cytotoxic and the helper T cells. And basically in allograft rejection, the major culprits are the CD8 positive cytotoxic T cells. Okay, the foreign MHC proteins, they are more potent inducers of T cells than the foreign non-MHC proteins. So MHC proteins of the donor and the MHC proteins of the recipient, their compatibility should be there, otherwise the graft will be rejected. Okay, so the stimulation of recipient's immune system by foreign MHC proteins, what is happening there, that antigen presenting cells of the graft in association with their MHC class 1 and class 2 proteins, they will activate the recipient's immune response. And the second possibility is that APC of graft present the recipient's proteins and they activate the recipient's immune response. And the third possibility is that donor's proteins and class 1 and class 2 proteins, they are taken up by the recipient's APCs and they activate the immune response. So these are the three possible ways that how the recipient's immune response system is evoked by these foreign MHC proteins. Okay, now allograft rejection is also of three types: the hyperacute, acute, and the chronic. The hyperacute means that the immediate rejection of the graft, and it is because of the you can say that preformed anti-ABO antibodies, means you can say that the preformed antibodies directed against the donor tissue, and this is caused by the accidental ABO blood type incompatibility, which is obviously very rare, and this even presents while still in the surgery with thrombosis and occlusions of the graft vessels. And it is, if I say that which hypersensitivity reaction is involved, it is associated with the type 2 type of hypersensitivity. Then we have the second one, which is the acute allograft rejection. It takes about weeks to months. Okay, it's a slower process, and this is a T cell mediated immune response, and this is, you can say that it is directed against a foreign MHC. Inflammation, and you can appreciate the inflammation and leukocyte infiltration of the graft vessels, and this is the most commonest type of allograft rejection. Then we have the third one, which is of the chronic allograft rejection, and it will take about months to years, and this is also a T cell mediated process, but it results from the, you can say that, foreign MHC, like looking like a self MHC carrying an antigen, and it results in the intimal thickening and the fibrosis of graft vessels as well as graft atrophy, and it is associated with usually type 3 and type 4 hypersensitivity reactions. Okay, now we have another condition which is known as the graft versus host disease. Okay, in previous slide I have shown you the allograft rejection, but this is a specific type of rejection which is seen in the 2/3 of the patients which have undergone the bone marrow transplantation. And its onset, obviously it varies, but it is a condition in which the donor marrow, means the graft which is taken from the donor, it attacks the tissues of the recipient, the host. So this attack is mediated by the T cells, and what will happen that T cells attack, and as I have told you that they if they are not able to differentiate between the self and non-self, what will happen that they will start killing them. And in this disease, the donor T cells, it recognizes the host cells as non-cells, and because the immune system of bone marrow transplant recipients is compromised by the disease, host tissues are you can say that they are not able to mount an immune response against the donor cell. So eventually what is happening, that the graft which is taken up by the body, it start destroying the recipient cells, and the donor T cells in the graft, they start proliferating, and they attack the recipient tissues. And the patients, what happened, that normally the patients are coming up, the bone marrow transplant patients are coming up with the diarrhea, rash, and the jaundice. In students, the onset of this graft versus host disease, or even the type of allograft rejection, is very important, because by this time period you can appreciate that whether the person is going to have that hyperacute rejection, or the patient is having chronic or acute. Okay, so what are the requirements that how the GVH reaction will begin? It means that the graft must has a immunocompetent T cells. Okay, the person should be immunocompromised, because normally we are giving the medications and the treatments so that they can suppress the immune system of the host. And the third requirement is that the donor T cell recognize the recipient cells as foreign. Okay, so this was all about allograft rejection, that the person is not accepting, and the body is evoking the response against this and this. But now the question comes that the fetus, the developing embryo in the womb of a woman, why it is not rejected, although this is also an allograft? Because why it is an allograft? Because the mother is having the baby with the genes inherited from the father as well, from the father and the mother, but still the body is not rejecting that graft. So there are different you can say that possibilities. You know that the mother formed antibodies against the foreign paternal MHC proteins, so this is the reason that the mother is not exposed to the fetal antigen. Okay, and the one possible reason is that the trophoblast layer of the placenta, it does not allow the maternal T cells to enter the fetus. So that is why this could be the reason that the fetus is not rejected by the body of the mother. Also, the trophoblast is not expressing the typical MHC1 and MHC2 protein. Instead of that, it expresses the non-classical MHC, and the non-classical MHC does not evoke the specific type of reaction which is required to reject an allograft. Okay, now we have understood the concept of MHC. Now we have learned about the transplantation and its type, and I am constantly saying that mismatch grafts are rejected. But how we are matching, how we are analyzing that this graft is matched between, there is a good match between the recipient and the donor, and this is all taken up by the processes which are performed in the laboratory. And these processes which are used to find out the closest match between the donor and the recipient is called HLA typing or tissue typing. So basically there are three methods which are used for HLA typing. The one is the DNA sequencing. This is the gold standard, and it gives very reliable and accurate results. Then we have serological assays. They are also important, and their sensitivity is about 80 to 85 to 90%, even 95%. And then the third one is a mixed lymphocyte culture, which is usually not performed if the DNA sequencing and the serological assays have given a satisfactory result. Okay, so the first one is the DNA sequencing. This is performed by the polymerase chain reaction. The DNA is amplified in different wells by using different sequences and primers of the different alleles. If the matching is there, then what will happen? The amplification will begin, and amplification of the DNA will begin, and we will be able to see the DNA on the electrophoresis. Gel electrophoresis on agarose gel. But if the mismatch is there, if there is mismatch, the protein amplification will not there, the DNA amplification will not there, and eventually we are having nothing. So the DNA is the sequencing is the gold standard method. The second one is the HLA typing. In this condition, we have to introduce different allo antisera, means that the serum which will be containing the antibodies anti-HLA, anti-HLA-A1, HLA-2, HLA-3, and so on. So what will happen that in the serological assays, the cells from the donor and the recipient, they are reacted with the battery of antibodies, means a set of antibodies, and each this antibody is specific for a different class of class 1 and the class 2 protein. Then in the next step, a complement is added, and those cells which contain the MHC protein, they homologous to the known antibody, they will lyse. And this is a good method. You can say that overall it gives satisfactory result about 90 to 95%. But again, if HLA typing is not giving you the satisfactory result, we can move to the another test which is known as the mixed lymphocyte culture or mixed lymphocyte reaction. What happens in this test that we have two type of lymphocytes. The one group is known as the stimulator lymphocyte, and the second group is known as the responder lymphocyte. The stimulator lymphocytes are those lymphocytes which are taken up from the donor, and the responder lymphocytes are those which are taken up from the recipient. The both stimulator lymphocytes and the responder lymphocytes are incubated and in cell culture, so that what will happen that the DNA synthesis will start happening. And this DNA synthesis, so we have to measure the DNA, that how much DNA is synthesized by the cells. So for that we have to add up the tritiated thymidine. So what will happen that this tritiated thymidine will bind to the newly synthesized DNA, and which can be easily measured. But what should be the ideal condition? So how can we say that this is a matched, means donor and recipient classes 2 and MHC proteins are not similar or similar? So this is measured by the amount of DNA synthesis. If a large amount of DNA is synthesis, it means this is a poor match, and if a small amount of DNA is synthesized, it means that this is the good match. So the best donor is the person whose cell trigger the incorporation of the least amount of tritiated thymidine on the recipient cells. So we have another very important test which we should perform, and this is known as the cross matching. What happens, because this is important, that if you are not cross matching, or then obviously you are going to make the person prone to the hyperacute rejection, because hyperacute allograft rejection is because of the preformed ABO antibodies. And by the way, the hyperacute rejections are also known as the white reaction, because at that time immediately there is an occlusion of the vessel. So what will happen in the cross matching? We have to take out the donor serum, and then we have to take up the recipient's RBCs. So the 2 drops from the donor serum and 1 drop from the recipient RBCs, they are incubated at the at 37 degrees centigrade for about 1 hour. And then what will happen that this reaction is centrifuged and anti-human globulin is added, and then again waited for some time. If the agglutination is there, it means that it is not compatible. Okay, but if there is no agglutination, it means it's compatible, and the cross matching is giving the result that there is a compatibility between the donors and the recipient. But student, this is the cross matching and HLA typing is a very technical process, and therefore to find a suitable donor is not an easy thing. Okay, so this was about the HLA typing in the laboratory by DNA sequencing, by you can say cross matching, and by serological assays. So this was all about the MHC.